Good afternoon, everyone. I'm Malika Ashford, an editor at Genomics, and I will be your moderator today. The title of today's webinar is Bringing a New Standard of Clarity to Solid Tumor Research with Single-Cell Genomics, and is sponsored by Mission Bio. Our speakers are Jorge Reis-Filho, Chief of Experimental Pathology Service at Memorial Sloan Kettering Cancer Center, and Julia Guffon, Translational Applications Liaison at Mission Bio. You may type in a question at any time during the webinar. You can do this through the Q&A panel, which appears on the right side of the webinar presentation. If you look to the bottom tray of your window, there are also a series of widgets to enhance your webinar experience. With that, I'll turn it over to Dr. Reis-Filho.
Thank you so much. It's an immense pleasure to be here with you today for this webinar. I'd like to thank Julia and the Mission Bio team for the opportunity of being here. I'm the Director of Experimental Pathology at Memorial Sloan Kettering and also the Director of the Experimental Pathology Fellowship Program. In this talk, what I'd like to do is to discuss with you single-cell DNA sequencing. So we'll not be focusing on other single-cell sequencing methodologies today, but single-cell DNA sequencing, and trying to make some sense of how the technology has developed over the last 12 years. So in this talk, I'm going to start first with a discussion of intratumor genetic heterogeneity and the approaches that we in pathology use to assess intratumor genetic heterogeneity, and then move on to my favorite technology of all here in this context, that's the single-cell DNA sequencing, and then recap with you all the developments coming from the first generation of single-cell DNA sequencing methodologies and how much we have learned through the cataloging of intratumor genetic heterogeneity using these approaches, how much we have learned in terms of the development, progression, and evolutionary trajectories of tumors based on these methods. Then I'm going to move on to the novel approaches for single-cell sequencing using direct library preparation and micro-droplet-based single-cell DNA sequencing with a study that's unpublished from our group on the development of a novel approach for us to be able to do this type of micro-droplet single-cell sequencing from frozen solid tumor samples, not only from fresh. So I think now in 2022, we have all come to terms with the fact that cancers are composed of mosaics of tumor cells that, in addition to the founded genetic events that are present in all cancer cells, they harbor private genetic alterations as well. And there are now many lines of evidence to demonstrate that some of these private genetic alterations mediate some of the biological phenomena that have baffled us for years, such as progression from in situ to invasive disease and from invasive disease to metastatic disease, as well as therapeutic resistance. In a way, the data that has emerged over the last 15 years is that cancers constitute let's say, a system whereby it recapitulates the other median evolutionary model where there is cooperation and competition between different clones within a tumor. And understanding this ecosystem, even from the perspective of the cancer cells only, is something that has informed us immensely. And there have been many approaches for us to study, catalog, and dissect intratumor genetic heterogeneity in tumors. One of the first approaches to be developed was by using bulk sequencing and then subsequent clonal decomposition. In an approach like this, one would perform whole exome or whole genome sequencing, and then on the basis of the variant allelic fraction, local copy number, ploidy, and tumor cellularity, one would infer what's called the cancer cell fractions, meaning the bioinformatically inferred percentage of tumor cells harboring a given mutation, and then using advanced mathematical methods, perform some clustering of these alterations to define the clonal clades within a tumor. This approach has proven really successful for us to understand the clonal architecture of tumors, but at times it's really hard when tumors have both copy number and mutational intratumor heterogeneity. It becomes really hard to deconvolute these cancers. The second approach is based on multi-regional sequencing, something pioneered by Charlie Swanton's group, where basically investigators would obtain multiple areas of a tumor, distinct areas of a tumor in metastatic deposits, perform sequencing analysis of each one of these anatomically distinct areas separately, and then do a joint analysis to understand the mutations that were truncal, the ones that were branch mutations, and come up with beautiful deconstructions of the phylogeny of a given tumor, and understand the genetic evolutionary, let's say, convergence and the acquisition of specific therapeutic resistance alterations. Then the development of methods for in situ detection of genetic alterations, including mutations. One of the first endeavors in this field was pioneered by Nelly Poliak's team, and I had the pleasure of contributing to this study, where they developed starfish that allowed for the detection of mutations in situ, and then understanding the distribution of these mutations within the tumor bulk. Now, many companies have developed approaches to detect mutations in situ based on DNA or RNA, and this is an evolving field. But I think the one that has had the most profound impact on our understanding of intratumor genetic heterogeneity has been single-cell sequencing. And here, it was a field developed by Nick Nevin and Jim Hicks back in the early 2010s. The principles of single-cell sequencing at that time, and now we may look at them and say, oh, they're quite simple, but at that time they were truly, truly groundbreaking. And basically, the way it worked was one would get a tumor or cell lines or a model, dissociate these cells, then obtain let's say a single nuclei soup that was put through a flow cytometry analysis, where the cells would be sorted on the basis of nuclear shape and DNA content, and then sorted one nucleus per well on a 96-well plate. The cells would be lysed, the DNA extracted, subjected to whole genome amplification. Then libraries would be prepared, and by multiplexing all of these cells, because once they were sorted here in the plates, they were barcoded, and then one could derive either copy number information or perform a mutational profile. And why do I say or? Because depending on the whole genome amplification methods, the data generated would be more amenable for copy number analysis or mutational analysis. And on the basis of this type of methodology, major discoveries have been made. For instance, here, coming from Nick Nevin's team, but through the analysis of breast and colorectal cancers, he observed that in breast cancers, there was a great deal of intratumor genetic heterogeneity within the tumors in the copy number space with different clonal clades. And an important observation that was made is that copy number alterations developed rather early in the tumor development, and that there was a suggestion that copy number alterations would evolve in the form of punctuated bursts of evolution, meaning they would be acquired in a stepwise manner rather than a gradualistic manner. And this was much better addressed or in a much greater detail addressed by Nick Nevin's team again in a subsequent study where they developed a method called Nuclei, again based on the same principles, but from a similar, from the same population of cells, they could derive mutations and also copy number information. And on the basis of this type of joint analysis, they could define the patterns of clonal evolution within breast cancers. They analyzed one ER-positive and a triple-negative breast cancer, and the conclusions that they drew at that time remain correct now, in that the copy number alterations doesn't matter if it's the estrogen receptor positive or the triple negative. The copy number alterations here highlighted by these numbers of chromosomes in the chromosomal arms happened early in the evolution of the tumor, and that these copy number alterations, when acquired later, where they were acquired in punctuated bursts of evolution. You can see here on the right. Whereas the mutations were acquired in a gradualistic manner, so in gradual steps throughout the evolution of the tumor. Importantly, they also made a very interesting and important observation here in that some of the tumors may really be polyclonal at the time of diagnosis, with multiple different clones constituting the final product of a tumor. They studied also triple negative breast cancers in detail using similar methodology, and they demonstrated that within triple negative breast cancers, again, in a larger cohort, the acquisition of copy number alterations as defined by single cell sequencing was in the form of punctuated bursts of evolution. And this information then could be really useful for us to understand clonal compositions within a tumor, given that there are not too many transitional forms. Single-cell sequencing methods were also applied to the study of disseminated tumor cells. In this study led here by Lise Born-Sandale's group in Norway, they studied primary breast cancers, lymph node metastasis, and then disseminated cells in the bone marrow using a very similar approach of a single-cell sequencing, DNA sequencing, and demonstrated that, of course, the disseminated tumor cells harbor the founded chromosomal alterations present in the tumor, but some of the disseminated tumor cells were more similar to the primary tumor cells. Others were more similar to the cells present in lymph node metastasis. But and demonstrated also that these disseminated tumor cells may be undergoing evolution with some form of geographic isolation, so a parallel evolution as well. Our group, when we entered the single-cell sequencing space, we had an interest in studying the progression of breast cancer. My team is dedicated primarily to the study of breast cancers, so we had one question in relation to the progression from ductal carcinoma in situ to invasive ductal carcinoma. And as you can imagine, it's really hard, it was really hard for us to be able to get on the same section different lesions, phenotypically different lesions that we could sample accurately by using fresh samples or frozen samples. So a postdoc in my team at that time, Luciano Martellotto, decided to embrace the challenge of developing an approach for single-cell sequencing analysis from formalin-fixed paraffin-embedded tissue sections. And when he proposed this to me, I laughed. I said, this is not going to work. But kudos to him because he did develop it. And we published in 2017 in Nature Medicine this protocol. But basically, we borrowed a lot from what Jim Hicks and Nick Navin had developed. We basically used from FFP samples. We extracted the nuclei using a combination of enzymatic digestion and physical forces, did this, the flow cytometry sorting. Then the step that actually changed everything in this regard was the introduction of a pre-whole genome amplification step. Basically, Luciano borrowed from DNA forensics some kits for DNA restoration. And upon using these DNA restoration kits, actually we were able to get high fidelity whole genome amplification using .PCR that then enabled us to perform the whole genome amplification, do the library preparation and multiplexing, and obtain rather reproducible and robust copy number data. And in our study in 2017, actually, we then analyzed ductal carcinomas in situ and their progression to invasive ductal carcinomas, and we posed the question of whether we could identify examples whereby the DCIS had a great deal of genetic heterogeneity, and in the process of progression from DCIS to invasive, there would be clonal selection. And this is one of the examples that we published in that Nature Medicine paper, whereby we have here in each column a cell. The chromosomes are represented from chromosome 1 to chromosome X here on the y-axis. My slides appear in a somewhat cut here. I do not know if you can see the bottom of the slide, but suffice it is to say that in yellow we have the cells that came from the DCIS, in green the cells that came from the invasive tumor. And remember, this is all from an FFPE section that we cut very thick and remove the components separately. And I hope you can appreciate here that there are many alterations that are actually clonal throughout the cells from the DCIS and invasive, but there were some alterations, some copy number gains and losses that were restricted either to the DCIS or part of the DCIS and the invasive. And when we did the clustering analysis, what we observed was that the majority of the DCIS cells clustered together and separately from the invasive component. And, but there was a subset of the DCIS cells that clustered together with the invasive component and actually was the most parsimonious origin of the invasive tumor. And here you can see the phylogenetic tree that we could draw up based on the copy number alterations in this tumor. And from the DCIS to the invasive, I hope you can appreciate here, that was a minor subclone of the DCIS that gave rise to the invasion. And in the process of the progression, there was a homozygous deletion of P10. And after the P10 has been linked to this process of the progression from DCIS to invasive. And after invasion, there was subsequent evolution. Importantly here, in this process, after the publication of this study in Nature Medicine, Nick Navin also studied DCIS, and other groups have studied DCIS, including the Precision Consortium, and they have demonstrated that the progression from DCIS to invasive is more complex. In some tumors, we do have this clonal selection. In others, there are multi-clonal progression. And some DCIS and invasive cancers are not even related. So there's much more for us to learn here, but single-cell sequencing enabled us to study this in sufficient detail. Now, so in summary here, what have we learned with this first generation single-cell DNA sequencing approaches? Actually, these single-cell sequencing DNA approaches actually provided several advancements for us in terms of the direct assessment of genetic alterations from single cells without the need of inferences coming from rather complex bioinformatics and computational biology methods. We could combine the sampling of phenotypically different areas with the single-cell sequencing, even by using laser capture microdissection. For the constitution of phylogenetic trees and inferring the progression of a tumor, very limited inferences were needed because we could obtain the data directly from the single cells. And with the development of the methods for both frozen and FFPE, and here Luciano Martellotto, Tumor, Baslan, Nick Navin, and many others played such a pivotal role in the field, but the archives have been unlocked, so many different types of samples could be utilized. The challenges that we faced with the first generation of, let's say, methods was that because of the whole genome amplification needed, there was an inherent level of noise. And the number of cells that were analyzed in those original studies was quite small. And given the need of flow cytometry, in fact, sorting for the nuclei to be put into the different wells, there were potential biases on the basis of the nuclear shape and morphology when we were performing those experiments. And for most of the methods available at that time, we could derive either copy number information or mutations because of the need of whole genome amplification methods, and because each one of these methods would be more amenable to either copy number analysis or mutation analysis. Now, in the 2020s, we have seen the birth of novel approaches for single-cell DNA sequencing, and they are available from academic groups and or commercial vendors. There are tens of different methodologies or variations of methodologies available. Of course, one of the methodologies is the one that I've just described to you, what I call the first generation. But now we also have technologies based on microdroplets, where basically individual nuclei are put into microdroplets, and then within these microdroplets, we can add all the reagents needed for us to barcode and amplify either the whole genome or specific regions of the genome. And there are also the methodologies based on nanowells, where basically one spots individual cells into nanowells, then performs all the reactions in this nanowells, and after that, with the barcoding in place, one can actually multiplex these reagents and these samples and perform a library prep and sequence. So in terms of the methods available, one that I find really exciting is direct library preparation, something pioneered by the groups from Reis-Filho and Saurabh Shah, where basically they initially used a microfluidic platform with a rather elegant microfluidic device that would create chambers to isolate individual cells that then would be barcoded and manipulated as needed. But they moved on to a different approach for this direct library preparation, where basically they use single cell spotting using a spotter, like a printer, spotting each nucleus in each of those nanoreactant chambers or those nanowells. And the principle of this method is really interesting, because upon having the cells in these specific chambers, what they do is to use transposase 5 to do tagmentation, basically to create the library for each one of the individual cells. They chop the DNA, the transposase chop the DNA and adds the barcoding in the same step. And by doing that, one enables the ability of doing whole genome analysis after multiplexing all of these barcoded cells. And it's a really powerful approach, because even with limited sequencing, let's say 0.5 whole genome equivalents here, one can derive from each cell sufficient copy number information that can be ascribed then back to each one of the cells after pulling the library with all the barcodes. And one can basically obtain copy number information in a genome-wide manner for each one of the individual cells. And then these clones, on the basis of copy number analysis, multiple cells can constitute a clone. Then these cells are merged, and one then can do mutation calling for specific subclones. So at the same time, genome-wide information for copy number and mutational data for specific clones within a tumor. And this methodology is really powerful then to reconstruct the clonal architecture of a tumor, infer clonal progression, and even derive clonal fitness. In fact, this approach has many advantages, including the fact that a really large number of tumor cells can be analyzed in one go. It provides genome-wide assessment. No whole genome amplification is required because the libraries are created with this transposase. It offers high fidelity copy number information at the single cell level and mutational data at clone level. And Saurabh Shah's group has done an absolutely phenomenal job in developing bioinformatics tools to assess this type of data, including tools for clonal decomposition, clonal fitness, encoded evolutionary trajectory of tumors, and even competition or collaboration between clones. The challenges that this approach faces is that it does require suspensions of nuclei, and the nuclei need to be really intact. The other point is that for the single-cell mutational profiling, then one needs to generate higher depths of whole genome sequencing, and then the prices currently are quite expensive. However, with the novel sequencing technologies, and there was a lot on in the press recently about novel sequencing methods reducing the cost of whole genome sequencing. So there's an opportunity here, perhaps, for this technology to be even more widely adopted. But for the interest of mutation analysis, if we were able to get to the single cell level, we face the problem of having some dropouts of genomic regions when we perform the documentation for library preparation. Now, another approach that I mentioned is the microdroplet single-cell DNA sequencing. It's something that is done by Mission Bio, and we have been using their technology now for 3 years. Basically, the principles are quite simple. After making this type of soup of nuclei, the cells are put through the device where they are then included into these microreactor chambers, these oil emulsions, where cell and protease are added. The nucleus and is in the microreactor together with protease. The cells are lysed, and then subsequently one adds all the reagents needed for the amplification of specific genomic regions to be performed, and at the same time the addition of the barcodes for each one of the cells. After that, with all the DNA amplified and barcoded, these can be merged together for a library preparation and subsequent sequencing. This technology actually has been really transformative in the field because there's no requirement for whole genome amplification. It can provide information in terms of both mutations and copy number, and the dropouts of the regions of interest that are amplified using those specific primers, the dropouts are quite low. And this technology has already been shown to provide really useful information from the perspective of hematologic malignancies. Ross Levine's group has published this wonderful paper in 2020, and we had the opportunity also here to collaborate with Dr. Pirolito at Memorial Sloan Kettering, where we performed the single-cell sequencing analysis of cell lines that he had generated to understand the mechanism of resistance of for KRAS G12C inhibition. This methodology so far has worked really well in the context of hematologic malignancies and cell lines, but there are many challenges that we face, that a large number of cells is needed as well, because we start with, let's say, 100,000, even more cells to end up having around 2 to 10,000 cells analyzed in the end. A priori knowledge of the genetic alterations is required because we need to find the genomic regions of interest that we want to sequence. And until recently, the use of solid frozen samples from solid tumors was quite difficult because we needed those soups of nuclei that were really intact. So to address this issue in partnership with Britta Weigel's team in experimental pathology at Memorial, and a study led by Arnolda Cruz Paula in our teams, we sought to develop an approach for us to be able to perform this single-cell microdroplet type of analysis from frozen samples. So the method that Arnaud had sought to develop was based on the principles of maximizing the number of cells that we could extract, so we could use minute biopsy tissues while enriching for tumor cells. So we had to be able to direct our sampling towards the tumor-rich areas and reduce at the same time the amount of nuclear debris and droplets. To do that, basically, then we obtained frozen blocks where we cut an H&E, found the areas of interest, then use a biopsy puncher to get those areas, and then using a combination of physical forces and enzymatic digestion that has to be adjusted to the tumor type, we could generate soups of nuclei that actually were intact. And after performing the visual inspection of a subset of the nuclei retrieved, where there are no clumps or no debris, we put it through the Mission Bio system to derive all the information that we need, including genotyping, copy number, allotyping, and even allelic-specific copy number information. In parallel with that, we needed to develop a system for us to analyze the data. And Mission Bio has been, a great partner in this regard, because we joined forces here and come up with an approach that borrows a lot from their pipeline, getting from the FASTQ file through several steps of the analysis. And then after getting the BAM files, we have developed several approaches for the variant calling, then the removal of noise here, and a technical noise. And then we wanted to do something else, not only go back from the, let's say, the bulk data available for us to see if those mutations could be found, but we wanted to do the Novo mutation calling from the single cell sequencing results. And to do this de novo mutation calling, even with the, let's say, smaller footprint of the single cell panel, we developed several filtering steps through the analysis of normal cells that were deployed and without known mutations that had been sequenced before, as well as several controls. And by employing this different combination of this complex combination of filtering steps, we were able also not only to do single cell genotyping, but also derive allelic-specific copy number information, as well as a single cell allelotyping for specific mutations. And we benchmark this against admixtures of cell lines, where we basically got the two pure cell lines, Ishikawa and HEC6. These are two endometrial cancer cell lines. And we sequenced them by single cell sequencing and high depth bulk sequencing. So we split the cells into one bulk, one single cell. And then we made admixtures of these cells. And then we could see what would be the limit of detection of specific subclones or subclonal mutations within these admixtures. And we concluded, after much validation, that we could identify clones with a frequency of 1% or more, with a really high I-square concordance between the bulk sequencing and single-cell sequencing. We next applied this method then to study some frozen solid tumor samples, and we chose endometrial cancers to do that for two reasons. First, that these tumors can have very different levels of mutations because they're the copy number low, the copy number high, the MSI high, and the poly-endometrial cancers. And they have different levels of copy number alterations as well. So here is an example where we performed single-cell sequencing of copy number low endometrial cancer. You can see here the results of the bulk sequencing with cancer cell fractions inferred, and then the results of the single cell sequencing from the same tumor. When we compare the VAFs obtained by tumor bulk analysis or a pseudo bulk using the single cell sequencing, you can see here that the R value was 1. So there's perfect agreement in terms of what we could derive. What this single cell sequencing analysis revealed to us was on two different fronts. One, we found that in the progression, or let's say in the development of this tumor, there was the acquisition of loss of heterozygosity of KRAS, where the wild-type allele of KRAS was lost, a phenomenon that has been reported to increase the oncogenicity of KRAS mutations. The second point is that in many endometrial cancers, we find multiple mutations affecting the PI3K pathway. And then we could investigate whether these mutations occurred exactly in the same cells, and if so, whether they were in cis or trans. And as you can see here, in this example shown here on the right, the mutations were actually found to be in the same cells and in cis. And the level of granularity that we can derive here is really, I find really fascinating, because then we can start probing these questions directly. This is another example where we analyze a microsatellite instability high endometrial cancer. And again, we performed the bulk sequencing and the single cell sequencing. And there were some clonal mutations here, the first 5 mutations, and then some clonal mutations by bulk sequencing. And as you can see here, the variant allelic fractions by single cell sequencing matched pretty well the variant allelic fractions by bulk sequencing. But it's really interesting. Look at these two mutations, PIK3R1 and RID1A. They have similar variant allelic fractions. And one of the potential inferences that would made from the bulk sequencing is that they would co-occur in the same cells. But single cell sequencing analysis revealed that these two mutations affected different clones. And actually, when we constituted the fish plot of this tumor, we could see that the clones were separate. And again, this type of granularity that single cell sequencing offers unlocks many opportunities for us to understand the evolutionary trajectory of a tumor. But I think the most exciting one from our study was the analysis of this poly cancer. And it was one of those endometrial cancers where we have a polyexonuclease domain mutation together with an MSI high phenotype, because the tumor had MSH inactivation as well. So we sequenced the bulk and the single cells. We did a comparison of the bulk and the pseudobulk by single cell sequencing, a very good R value again. And we could reconstruct the phylogenetic tree of this tumor. And as you can see here, this is a cancer where there was clear parallel evolution, with some of the branches being driven by different PI3K type of alterations. So a convergent evolution by the co-option of different components of the pathway. But given the number of mutations, because it's an ultra hypermutated tumor, so we wanted to understand whether we could infer what would be the mutagenesis processes operative in these late clones within this endometrial cancer. So what we did then was to get the mutations that were present in the bulk sequencing and in the single cell sequencing by overlaying the bed files of the two. And then we did a mutation signature decomposition analysis of the mutations present in bulk and the single cell, or detected only by single cell sequencing in our de novo mutation pool, and consistent with our hypothesis. In this case, when we performed the clonal decomposition of these nascent single nucleotide variants, they had the mutational signature that we'd expect to see in this tumor. That is a signature 14. That is the signature characteristic of endometrial cancers with both poly and MSI high. Confirming here that one can go from a solid tumor sample to the de novo mutation calling in minor subclones using this type of approach that is offered by Mission Bio. So in conclusion, I hope I have demonstrated to you here that single-cell DNA sequencing can offer many potential opportunities for translational research, including understanding the evolutionary trajectory of cancers, understanding also the mechanisms of therapeutic resistance. If we think about that parallel evolution, how much granularity would have to study questions of that nature, investigate clonal dynamics, in addition to doing clonal decomposition, understand the progression of tumors, and even tracking clonal evolution in this way. I find it really appealing also the idea of monitoring response, on the basis of single cell sequencing analysis of circulating tumor cells. It's still a bit challenging because of the number of cells required, but it's a field that many are focusing on at the moment. The other aspect that I find really exciting, and Julia will allude to in her talk, is the potential of these single-cell methodologies for one to perform not only single-cell DNA sequencing, but do this multi-omics type of approach. And this idea of combining protein analysis with single-cell DNA sequencing is really exciting. And our laboratory is now working on methods to be able to obtain intact single cells from frozen samples. It's still a bit of science fiction, a bit challenging, but it is something that we are working on. With that, I'd like to thank you for your attention. I'd like to acknowledge our funders here and actually the people who did the work, namely Britta, Arnaud, Ingi, and Shirin, and of course, Luciano Martellotto and Charlotte Ng, who actually did all the development of the FFPE methods for single-cell sequencing. With that, I would like to pass it to Julia Gouffon, who is the translational application liaison from Mission Bio. We'll tell you more about Mission Bio, and I'll be delighted to take questions after her presentation. Thank you.
Mm-hmm. Okay, great. Can you hear me? Hopefully, yeah. Okay, thanks. I appreciate Dr. Reis-Filho for working with us today and presenting that great history of single-cell genomics. Full disclosure, he is our Center of Excellence for solid tumor work at Memorial Sloan Kettering, and we're very, very happy that we have the ability to partner with him. So let me see if I can get this to advance. All right, here we go. So I'm going to just introduce a bit more of the Tapestry platform and specifically for our solid tumor research, but also throw in a little bit about our heme program. So we did spin out of San Francisco, out of UCSF about in 2014, 2015, and we launched our very first Tapestry instrument in 2017. And we've grown very, very rapidly. I think in the last few years, I think I was employee 60 or something, and now we're approaching, I think, 200, and we have many users worldwide. But the primary thing that we're focused on doing is single-cell DNA sequencing, and then also combining the ability to look at cell surface markers to identify which types of cells have mutations in them. So that's performing immunophenotyping as well. And the importance of this, we feel, is that as many of you know, we have a single hit often that starts out the cancer. Oh, I forgot, you guys can't see my cursor unless I put this on. A single hit that starts out with the cancer, and then as the cancer evolves, we have multiple types of mutations, as Dr. Reis-Filho just showed us during the tumor evolution. And if you're doing bulk sequencing and combining all of these cells at the end, you're not able to identify perhaps that you've got a mixed population here. This were perhaps some subclones population at the outset that then become resistant to therapy. Or, you have some that have co-occurring mutations. So that's really what we have set out to do, both in the heme space and also in the solid tumor space, is the ability to, as soon as possible, find these so that we can perhaps work on combination therapies and knocking out these clones to prevent progression over time. And again, as he explained before, when we do the bulk sequencing, we're really using some kind of algorithms that infer what the clonal frequency is, and those can be ambiguous or misleading. Also, we are unable to kind of look at the tumor purity and wild type. We have to estimate that a priori ahead of time by using like H&E stains or something like that, and then applying that to see, what if it's 80% tumor burden or 20%, that affects the calculations. Whereas where we can capture that we do have wild type cells here that do not contain the mutations of interest that we're looking for. Also, when we design panels, we can look at the zygosity, so we can know if there's a heterozygous or a homozygous mutation occurring. And then we can look at co-occurrence, and then very rare cell detection. For our LOD in heme samples, we have a 0.1% for just a single SNP. But if a clone happens to have multiple SNPs, then that gives us more information, and that we can identify that that is a true signal occurring in those cell populations, and we can go down even lower. This is just essentially a table that just repeats what I just said. Things that are heterozygous, we can actually quantify their zygosity and the clonal architecture as well. So this is the instrument, what it looks like. We're able to, in solid tumors, look at SNVs, also with indels, arm level copy numbers, and also gene level focal amplifications. This all depends, of course, on the design of the panel, the primers we use, and where we place them. So we have field application specialists that help and assist with this, and are available for everyone. And this allows us to unambiguously identify the variant zygosity and mutational co-occurrence, and that detect rare cell populations. We are working with many partners other than Memorial Sloan Kettering. We have a core lab that can run samples at Johns Hopkins, for example. We have partnered with larger CROs like LabCorp to run samples, and we also have a pharma applications development lab that is in San Francisco for us to do proof of principle for pharma companies interested. So this is the entire process. The first, we design a DNA panel. We have some that are already pre-designed. For example, Ross Levine has a myeloid clonal evolution panel. We also have some solid tumors panels that we have developed in silico for breast cancer and GBM, and we have a tumor hotspot. This is the instrument. It sets up front of many different Illumina sequencers, so that's a key part of the cost, is which sequencer you're able to run on. We have this Tapestry pipeline and the insights that are cloud-based analysis tools that allow the basic user hopefully to be able to take their data. It's not necessary that you have to have a bioinformatician work with you, but it's useful if you have one. And this is how it works. Let me see if I can click one more time without advancing the slides. It looks like something's missing. But if you are interested in doing any kind of on-surface labeling, then that's done by using BioLegend TotalSeq D. D is in DNA antibodies. They have an entire hematopoietic differentiation cascade panel. Also another 80-something conjugated clones that have an oligo tag on the end of them, and that's what we use to read the sequences off of, and so we can enumerate those similarly to what you would do with flow cytometry, but we're doing sequencing read. And as George mentioned, if you're interested in this, because we are using so many washes for non-specific binding, you need like 1 million different ones. So, and then we have the encapsulation step, which is critical to differentiate us from other microdroplet platforms, as that linearizes the DNA so we can gain access in the second step to the barcoded, putting in the barcoded beads and then primers for the interrogate the areas of interest. We do about 60 to 80x coverage on each of those amplicons, and if we are going to be sequencing a protein library, we actually do a pull down and separate the protein from the DNA, and we recommend 500x on that. The end result then is going to be something very simplistically depicted here, but we can see the specific IDH1 mutations you have and IDH2 mutations you have in this cell population. And in this cell population, you have IDH1, NPM1, and TP53. Okay. What do we do? As I said before, we've got hematology. Many different groups have already prepared panels that are in our catalog, or we have published with them. We also have some virtual design panels. Virtual means we bioinformatically run those. For solid tumor, we have that tumor hotspot panel, and then we have a breast cancer panel and a GBM panel as well. We also have a nuclei isolation protocol, and then have partnered with S2 Genomics for instrumentation to prepare nuclei. And then another group here that we have in Mission Bio is cell and gene therapy. So they're tasked with partnering with people that are doing genome editing, so CRISPR edits, and using mouse models to make sure that you have exact targets when you're editing and not off targets that can be make complications if you're trying to derive a new therapy. So again, these are the panels that we have for solid tumor. This is the new breast cancer panel. It's available on our website if you'd like to take a deeper look, but as you can see, it has not only hotspots, but also focal amplifications and arm level. And recently at AACR, we launched our new nuclei isolation protocol. And this just shows the ability of our CAT tool, which is the chromosome analysis tool, to call these gains and losses at an arm level. And then also we're able to look at, BRCA, a loss there, and then we have a gain in EGFR. These are in these breast cancer samples. And we have an RB1 loss. You will see that we've got PIK3CA and perhaps KMT2C that may be amplified, but we need to add, some more probes there, or all the probes have to be going in the same direction for us to call those. So for our preprints, you can find all of these online. Mainly, it's looking at therapeutic resistance is in clonal heterogeneity is our main drivers. This is showing that we are partnered with S2 now. And this is just the last one I'm going to show before we can get started. This is Hoshin Zhang and Christine Yakabushio Donahue. They're also at Memorial Sloan Kettering, where they did this DNA isolation using the S2 instrument. And it's pretty easy because it takes your hands off. And I know Arnaud talks about the nuclei prep they've done in his lab where they're doing the frozen samples, and it's quite tedious to make sure that you can mince the samples quite enough. But this just shows the ability to look in at this clonal trajectory. This is kind of a fish plot where you can look at these primary tumors and see how they change over time and create these liver metastases by having a homozygous deletion of a SMAD4 region. And I'm going to skip this, I think, because George covered this in his paper. Our presentation rather with Pirolito and Yuli Zhao, looking at KRAS inhibitors to G12C, but you can see that this just allows you to see that there is, in different cell populations, this KRAS G12C, which was the founder mutation, co-occurring with a point mutation here in KRAS or BRAF or NRAS mutations. And that's all for my part, so I just wanted to open it up again for any kind of questions that anybody might have. And again, I thank the Precision Oncology Group for allowing us to present here today. So any questions?
Thanks so much, both of you. As a reminder to webinar participants, if you have a question, please type it into the Q&A box in the control panel. We'd also like to ask attendees to take a moment after the webinar has ended to take our exit survey to give us your feedback. We will now start the Q&A portion of the webinar. First question we have here is about liquid handling and automation solutions, around these kinds of explorations, either, to do the sequencing or handle the liquids and single cells, for the Mission Bio system, obviously there's the microfluidic, component itself, but what else do you have to do on either end, and if you're choosing other methodologies? Mm-hmm. George.
Yeah. George, do you want to answer that, or shall I?
Either one of you.
Julia, go ahead, and then I'll add some aspects from our lab.
Yeah. So currently we don't have any liquid handling. It's usually you can process with one instrument, we say up to 8 to 16 samples a week, but that's with a single person, and they may have 2 tapestries. But right now it is about 4 hours the first day, I think, hands-on time. That excludes the preparation of the nuclei. And then it's just, having it off on the PCR instrument for a little bit of time and then putting it back on into the second fluidic cartridge. So we don't have any liquid handling on that part. Now, once you create the amplicons, you may be able to put these onto a liquid handling system to create the Illumina libraries if you had one of those. Anything else, George, to add?
No, it's exactly the same point. The automation comes after the sample has been processed by the emission bias device. Then we can do some automation, but the steps prior to that, we're still doing largely manually. But they can be standardized and they are quite robust. So, but it's still somewhat labor intensive.
Mm-hmm.
Julia, is there a maximum size for the DNA panels? Can you hold exome? And what's the limitation there?
I don't know if we didn't stress this, but we're really doing this at scale. Dr. Reis-Filho mentioned this. Instead of just isolating, a 96 or a 384 well plate, doing where you do a droplet or flow cytometry, we are capturing, say we put 100,000 cells on, we're capturing 5 to 10% of those cells. So we're sequencing 7,000 to 5,000 to 7,000 or even 10,000 cells at a time. So that's equivalent of doing 10,000, whole exome sequences, which can get even with the new sequencers that say they can do things for $100, going to be pretty expensive. So our panel sizes, we can go up to 1,000 amplicons, but most of our designs, people have already done some kind of whole exome sequencing, and this is really more translational basis discovery. So they know what they want to look for, and they design specifically for that, which greatly reduces then the sequencing costs downstream.
Gotcha.
But the average is somewhere around 300 amplicons or even less.
And this is for George. Someone asks, how reliable is de novo SNV calling from the Tapestry data?
That's a wonderful question. Thank you so much. And this is something that we have spent quite a long time trying to understand the level of technical noise that is included in the data generated at the end when we're trying to do de novo mutation calling. And we realized that we needed to apply several filters, some of which were derived through the analysis of multiple normal cells deployed that came from several different sources for us to define the background error that was coming. And then we derived filters on that basis to apply to the samples. Once we apply those filters, the mutation calling was quite robust, and we could identify in the bulk sequencing, then not called because the frequency was too low, but reads supporting the same mutations for quite a few of them. So we're feeling more and more comfortable, but along with another question that was pasted in the chat, we still sequence at a number of normal cells per run, so that we have an internal control for the, let's say, the background level of SNVs that we detect in those normal cells, and use that to help us with our mutation code.
And are those normal cells coming from the sample, or do you have to use an extra normal control?
When we are doing tissue samples, one of the problems that we always face with the tumor samples is that tumors are not balls of cells, solid balls of cancer cells. There's always stroma, inflammatory cells, and so on. Given that we have information from the clonal truncal mutations from those tumors already, because we had sequenced their bulk, we use that information for us to identify the cells that do not have any of the mutations of the normal of the cancer cells, and we use that information as part of the pool of normals for that given sample. So it comes from the same sample as the tumor. For models, we always include a diploid cell line, at least some cells from a diploid cell line for the same analysis.
One more question we have from the participants is, if someone's conceptualizing how how to use these methodologies to check if a drug is targeting, KRAS G12C in vivo model. How do you do that? Walk us through.
No, one could use this for one to understand the shifts in the populations upon the challenging of the models or even the patient samples with the drug. However, these methodologies are not designed for us to define target engagement and target inhibition. It's a different set of technologies. We derive information here from the analysis of multiple time points after the intervention with the drug. And then it can be really informative for us to understand whether there's the acquisition of additional mutation in the same pathway. There is, let's say, an acquisition of alteration in a parallel pathway that's causing resistance. And we can do this as a single cell form.
All right, well, that is all the time we have for today. We'd like to thank Dr. Reis-Filho, Julia Guzman, and our sponsor, Mission Bio. If we didn't have time to get to your question, we will try to follow up with our experts. As a reminder, please look out for the survey after you log out to provide your feedback. If you've missed any part of this webinar or would like to listen to it again, an archived version will be emailed to all attendees. Thank you for joining us for this Genome webinar.
Great. Thank you. Bye.
Thank you.